Earlier, we were discussing regarding the N group analysis by using the identification of N-terminal amino acid residue. And here in these methods, we are going to determine the number of different polypeptide chains which are present in the proteins. Now we are going to discuss about the N group identification analysis by using the identification of C-terminal amino acid residue. And here we are going to discuss about the Akabori method, and second method is by using the enzyme carboxypeptidase. And here under the Akabori method, we are going to use the reagent hydrazine. And here in this case, the polypeptide chains are treated with the hydrazine at the 90 degrees Celsius for 20 to 100 hours in the presence of a mildly acidic ion exchange resin. And here in this case, all the peptide bonds are broken down, and here each amino acid is going to react with the hydrazine and going to form the amino acyl hydroxides. And here except the C terminal residue. And here in this case, the C terminal residue which is released as a free amino acid is then identified chromatographically. And as you can see here, so here this is the polypeptide chain, and here this polypeptide chain, it is going to react with the hydrazine at the high temperature, and here it is going to result in the breaking the polypeptide chain into free amino acids. And here all the amino acids are going to react with the hydrazines and going to form the amino acyl hydroxides except the C terminal amino acid, so which is released as the free amino acid. So here this C terminal free amino acid which is separated, and here which is identified chromatographically. And here the next method is carboxypeptidase method. And here in this method, we are going to use the carboxypeptidase enzyme, which is going to cleave the polypeptide from the C terminal end and removing the one amino acid at a time. Removal of one amino acid leaves behind a new polypeptide, which is the target for the carboxypeptidase also. And here in this case, this is the polypeptide chain, and here if it is exposed to the carboxypeptidase means, and it is going to cleave the C terminal residue which is indicated in the red color, and which is released, and it can be purified and identified. Okay, so here by using these method, we can able to perform the N group analysis. And under the N group analysis, we are going to determine how many different polypeptide chains are there in the protein by using the N-terminal residue identification, or by using the C-terminal residue identification. And here you may ask me one question: you can do either any one of these two methods. But here in this case, these N-terminal residues are the C-terminal residues. Sometimes they might be deeply buried inside the proteins. So here in that case, it will be difficult to perform these experiments. So here for our purpose, we need to perform the both C terminal and the N terminal amino acid residue for the correct results. So here, for example, one protein is there. If it is having the two polypeptide chains means, if it is going to reacting with the Sanger's reagent means, so here the Sanger's reagent, it is going to react with the these two amino acids which are present at the N terminal residue, and going to form the amino acid residue which is bonded with the Sanger's reagent. So here by doing so, we can able to identify the how many number of different polypeptide chains are there in the proteins. Okay, so here after the N group analysis, after the identification of the number of polypeptide chains in the protein, now these polypeptide chains has to be separated, and here it is going to be performed with the help of the cleaving the disulfide bonds, and different subunits are the polypeptide chains are going to link to one another with the help of the disulfide bonds. And here the purpose of cleaving the disulfide bonds is include. So here it is results in the separation of the polypeptide chain. At the same time, the protein is present in the native conformation, which is stabilized by the disulfide bonds. And here it is going to obstruct the action of the proteolytic enzymes which are used in the further steps. And here for this purpose, these disulfide bond which is connecting the different subunits are the polypeptide chains has to be cleaved. And see, by doing so, we are going to separate the polypeptide chains. And here how we are going to cleave the disulfide bonds. And here the disulfide bond, usually it is going to form in between the cysteine residues. So here the cysteine residue of the one polypeptide chain and the cysteine residue of the other polypeptide chain, which is under the free sulfidal group, are going to react to one another and going to form the disulfide linkage. And here this disulfide linkage, which is one of the important the linkage which is going to determine the protein structure. And here it can be cleaved with the help of the reagents such as beta mercaptan ethanol or dithiotherol or formic acid. And here in this case we are using the beta mercaptadinol. Beta mercaptadinol going to react with these disulfide bonds, and here it is going to reduces these disulfide bonds and going to form the free sulfidyl groups, and by doing so it is going to separate these two polypeptide chains. Okay, so here after the separation of these two polypeptide chains, it is going to form the free sulfidyl groups. And here these free sulfidyl group, it can once again react to one another, and it may form the disulfide bond again. And to avoid this one, it will be treated with the iodoacetic acid. And here this iodoacetic acid going to react with the free sulfidyl groups of the cysteine residue in the polypeptide chain, and going to form the S-carboxymethyl cysteine. And by doing so, here the disulfide bond will not be formed again. And here some of the non-covalent bonds which are present in the protein, it can be broken down at the extremes of the pH, at a high salt concentration, or in the presence of reagents such as urea or guanidine hydrochloride. And here yes, so here after this one of the important method under this one is we need to determine the amino acid composition of the protein. So here the identification and the quantification of amino acid in the polypeptide chain, it is one of the important step in the determination of primary structure of proteins. And here in this case before the turn the polypeptide chains are hydrolyzed into individual amino acids by the acid hydrolysis and the alkaline hydrolysis. Under the acid hydrolysis, polypeptide chains are hydrolyzed by using 6 normal HCl and heated at 110 to 120 degrees Celsius for 24 hours. And here in this method, some of the amino acids such as glutamine and the asparagine will be hydrolyzed to their corresponding acids into glutamic acid and the aspartic acids. And here it is going to liberate the ammonia. And here the measurement of ammonia produced gives the total amount of amides present in this sample. So here we can able to determine the total amount of amides present in the sample, but now we cannot able to determine the glutamine or the asparagine directly. And here the second method is alkaline hydrolysis. In the alkaline hydrolysis, polypeptide chains are hydrolyzed using the 5 molar NaOH. And here under these conditions, some of the amino acids such as cysteine, serine, threonine, it cannot be recovered completely. And here thus the results of both acidic and alkaline hydrolysis are required to determine the total concentration of all the amino acids present in the sample. And here after the acid or the alkaline hydrolysis, so here after the treatment, the individual amino acids whatever they are released, so all those amino acids are separated using the cation exchange chromatography. And here further the column eluate is further mixed with ninhydrin, which is going to react with the most of the amino acids and going to give the purple color, and the absorbance is measured at the 570 nanometer. And by doing so, we are going to determine the amino acid composition of the proteins. Thank you.